The Center for Modeling Immunity to Enteric Pathogens MIEP has just released an upgrade to the Enteric Immunity Simulator ENISI. This new modeling and simulation tool allows users to create enteric mucosal immune systems in silico with an agent-based software, providing a better glimpse into the mechanisms underlying host responses to pathogens invading the bacteria-rich environment of the gut. This tool will help immunologists to study mucosal immune responses to infectious and immune-mediated diseases. The MIEP program is a $10.6 million effort funded by the National Institute of Allergy and Infectious Diseases at the NIH. To access ENISI, from the home page, go to Modeling, Tools, ENISI. To run simulations, click on Simulation. To run simulations with the ENISI using the graphical user interface, the user specifies the size of the initial mucosal cell populations. These include, in order, epithelial cells, resting macrophages, tolerogenic bacterial strains of the lumen microflora, immature dendritic cells, resting CD4 positive T helper cells, resting natural T regulatory cells, as well as active T cells of various phenotypes Th1, Th17, and Treg in the lamina propria and the lymph node. The values given represent the immunological set point of the host as well as the tissue sample size. For example, in this scenario, initial cell numbers are set to represent an immunologically naive mouse and a sample size of 1% of the mouse stomach. The user then provides infection specifics by providing the dose of either inflammatory pathogen or commensal bacterial strain to be given on a specified day of the simulation. Inflammatory bacteria represent transient pathogen that elicits various inflammatory factors and may be eliminated upon contact with immune cells. Commensal bacteria is a strain that is present in the lumen microflora and may have both tolerogenic and inflammatory capabilities depending on the model specifications made as described in the ENISI version 0.9 documentation. In the ENISI model, only bacteria in association with epithelial barrier are represented. Hence, the dose of bacteria will not directly translate to the concentration of CFUs administered experimentally and may have to be estimated by fitting simulation output to available experimental data. In this scenario, we give a dose of 10 commensal bacteria on day 4, where parameter settings have been made so that commensal bacteria represents the H. pylori strain 26695. Finally, the user provides the length of the simulated infection, where one cycle equals 6 hours or 1/4 of a day. Once the simulation is complete, plots will be provided on the right-hand side. These show the counts of T cells in the lamina propria, T cells in the gastric lymph node, macrophages in the lamina propria, and damaged epithelial cells over the time course of the simulated period. To get a better view, click directly on the plot. Here we show a plot of T cells in the lamina propria over the time course of infection. In these plots, the x-axis is the simulation cycle that corresponds to 6 hours, and the y-axis is the cell count in the tissue sample. Here we see that there is a high level of Treg due to the presence of tolerogenic microfloral strains, and a low level of Th1 that is specific to H. pylori until after day 30, that is cycle 120. By day 60 post-infection, that is cycle 240, we see an obvious Th1 dominant response in the lamina propria. From this page, one may also view results with the interactive result viewer. This option allows one to view a schematic of the interactions taking place during the simulation. This is in the form of a network model of inflammatory and regulatory immune cell interactions with epithelial cells and bacteria in the 4 tissue compartments: lumen, epithelium, lamina propria, and lymph node. Red nodes indicate cells that participate in the inflammatory pathways, and blue indicate those in the regulatory pathways. Green nodes are cell types that may participate in either response. The selector on the upper left-hand corner of the screen allows one to navigate the network. One may zoom in and then click and drag in order to move around. The cell types represented are listed on the right-hand side. To identify what each cell type represents, one can click on the abbreviation. Here we choose M1 macrophages. Clicking on the abbreviation leads one to a window where a description of the cell type is given, along with a time course for the count of that cell type over the simulated infection and a reference. One may also access this information by clicking on the flag that is directly on the node. You will also notice that there is a flag for each tissue compartment. By clicking on the flag of a tissue compartment, this information is provided in the form of a heat map where the x-axis is the time point post-infection and the y-axis is labeled with cell types. For each cell type at each time point, the color indicates the cell type count in the tissue compartment. Red indicates the highest values for counts over all cell types, blue indicates the lowest, and white is the count in between these lowest and highest values. Here we can see that in early infection, the highest counts belong to resting and tolerogenic immune cells, with inflammatory Th1, Th17, and M1 increasing towards the end of the simulated infection period. Also on the right-hand side are the list of interactions in the model. Upon clicking on an interaction, one is located to the interaction in the network and is provided with a description of the interaction and a reference. To get this information, one may also click on the arrow representing the interaction. To view the results of various experimental conditions in silico, click on the Simulation tab from the main ENISI page. Here we provide results for simulated infection under hypothetical experimental conditions that have been pre-configured for past in silico studies. The release of ENISI helps the MIEP team to fulfill the goals of developing user-friendly and interactive modeling tools that engage and inform the immunology and infectious disease communities, thereby enabling paradigm-shifting scientific discovery. Such powerful simulations will ultimately accelerate the discovery of novel drug targets and biomarkers for enteric infectious diseases. The fully integrated computational modeling and immunology experimentation efforts within the MIEP program enable generating mechanistic evidence in silico and efficient validation in vivo.